In the year 2000, a geneticist ran a lab test that, according to the data, made two peoples who had spent decades defined by their conflict come back looking like distant cousins. What did that actually mean? The room where it happened wasn't a courtroom or a peace summit. It was Cold Spring Harbor, New York, at a genetics conference, and the researchers on stage were presenting Y-chromosome data live while their home region was gripped by active conflict. Somewhere in that presentation sat a number that nobody in the audience expected to hear stated so plainly. Over 70% overlap in a shared paternal gene pool. No context yet, no explanation, just the figure hanging in the air of a scientific meeting while the news cycle outside told an entirely different story. This isn't folklore, and it isn't political rhetoric dressed up in lab coats. It's peer-reviewed population genetics, the kind that goes through the same review process as any other study, published in journals like PNAS, Nature, and the American Journal of Human Genetics. The question this documentary is built around is a narrow one, and it's worth stating exactly. How genetically close are these two populations, really? And what does the DNA actually say, as opposed to what people assume it says once the word DNA enters a conversation already loaded with history? If population genetics rabbit holes are your thing, there's a lot more of this coming, so stick around and subscribe while you're here. Because the number itself is the story, there's no need to build toward it slowly. The 70% figure isn't the end point, it's the entry point. 3 separate research teams, working with 3 separate sample sets across more than one continent, ran the tests independently. And before we look at what any of them concluded, it's worth understanding why a question this old took until the very end of the 20th century to get a genetic answer at all, and why, once it arrived, it refused to stay confined to an academic journal. Jews and Palestinians both trace their identity narratives back to the same ancient region, the Levant, but they arrive there through very different modern histories. One carries roughly 2,000 years of diaspora, dispersal across continents, and eventual return. The other carries a continuous regional presence that never broke. 2 populations, 1 shared point of origin, and 2 radically different stories about how they got from there to here. That combination is exactly the kind of question population geneticists find irresistible. Competing origin narratives, enough historical documentation to actually test something against, and a biological record that doesn't care which side of a border it ends up on. The tool researchers reached for first wasn't the whole genome. It was uniparental markers, the Y-chromosome, passed from father to son, largely unchanged across generations, and mitochondrial DNA, passed from mother to child. Both act like a kind of genealogical fingerprint, stable enough to trace a paternal or maternal line back through centuries without the mixing that happens elsewhere in the genome. The earliest hint that something interesting might be sitting in that data came in 1993, when a team led by Santa Chiara Benerichetti suggested a common origin for Jewish and non-Jewish Middle Eastern populations. It was early work, done before high-resolution genotyping existed, more a signal flare than a conclusion. The technology of the time simply couldn't resolve the question with any precision. One boundary is worth setting now, because this documentary holds to it throughout. Genetic ancestry data describes population history. It does not settle political or legal questions about the modern conflict, and no study in this series makes that claim. Genes can tell you where people's ancestors likely stood on a map centuries ago. They cannot adjudicate a border dispute in the present day. What's surprising, looking back, is how long the wait actually was. Given how much scientific interest there was in the question by the early 1990s, the first real high-resolution study wouldn't arrive until the very end of the decade. That study landed in 2000, and it's still the one every later paper has to reckon with. Michael Haber led an international team that analyzed specific regions of the Y-chromosome in 1,371 men, drawn from 29 populations spread across the globe. The sample wasn't narrow. It included both Jewish and non-Jewish participants from the Middle East and North Africa, alongside comparison groups from sub-Saharan Africa and Europe, giving the study a wide enough lens to place any regional finding in a genuinely global context. What came back was specific. Jewish men shared a common set of genetic signatures with non-Jewish Middle Easterners, and the study named names. Palestinians, Syrians, and Lebanese populations all carried those same signatures. At the same time, that shared Middle Eastern pattern diverged significantly from the genetic profiles of non-Jewish men living outside the Middle East and North Africa. The line wasn't blurry. It separated cleanly along regional lines, not along the lines conflict narratives usually draw. Study co-author Harry Oster of the NYU Human Genetics program gave the finding its quotable line, saying that Jews and Arabs are all really children of Abraham. It's the sentence that made headlines, but it wasn't the only finding worth noting. A second result, far less publicized at the time, showed that despite thousands of years of diaspora and dispersal across continents, Jewish communities had largely not intermixed with the non-Jewish populations that hosted them along the way. Two findings, pulling in different directions, deep shared regional origin on one hand, and remarkable genetic continuity, despite centuries of separation on the other. The study was published in the Proceedings of the National Academy of Sciences in May 2000, timing that put it in front of a global audience during an active period of regional conflict. The authors weren't chasing a peace process talking point when they designed this research. They were testing a population genetics hypothesis, using standard methodology, and the signal that came back was cleaner and more specific than they had reason to expect going in. That was the worldwide comparison, built from 29 populations spanning several continents. The Israeli and Palestinian specific data came from a separate team, working an hour away in Jerusalem, with a narrower sample and a sharper question. Nebel's team published their findings in two stages: an initial paper in Human Genetics in 2000, followed by an expanded version in the American Journal of Human Genetics in 2001. The sample was tightly defined, 526 Y-chromosomes, drawn from 6 distinct populations: Ashkenazi, Sephardic and Kurdish Jews from Israel, Muslim Kurds, Muslim Arabs from Israel and the Palestinian Authority area, and Bedouin from the Negev. Rather than the broader markers used in earlier work, this team tested 13 binary polymorphisms alongside 6 microsatellite loci, a higher resolution method that could resolve finer distinctions within the data. The headline number is the one that gets quoted most often: 70% of Jewish Y chromosomes and 82% of Palestinian Muslim Arab Y chromosomes belong to the same chromosome pool. In the researchers' own words, the data revealed a remarkable degree of genetic continuity in both Jews and Arabs, despite their long separation and wide geographic dispersion. There's a nuance buried in that data that almost never makes it into the headlines. Roughly a third of the Palestinian-specific chromosomes formed a tight, closely related cluster of haplotypes that was rarely found among the Jewish samples. Those chromosomes carried J1 markers, possibly linked to a later migration originating from the Arabian Peninsula, a distinct thread within the broader Palestinian genetic picture that doesn't show up on the Jewish side of the comparison. A second nuance runs in the opposite direction. The same dataset showed Jewish populations trending slightly closer, genetically, to groups from the northern Fertile Crescent, among them Kurds, Turks, and Armenians, than to their immediate Arab neighbors. So the 70 and 82% figures, striking as they are, sit alongside finer-grained patterns that pull each population's story in its own particular direction. Move the lens from the Y chromosome to the whole genome, and the picture gets one layer more specific. At Hebrew University, geneticist Ariella Oppenheim led a study built around a more targeted comparison: the Y chromosomes of 119 Ashkenazi and Sephardic Jews, set against 143 Israeli and Palestinian Arabs. What her team found was that key DNA segments across many of these men were similar enough to cluster into just 3 haplogroups, out of the far larger number of haplogroups that exist across human populations. The microsatellite data, drawn from shorter, faster mutating DNA segments that make useful population clocks, pointed to common ancestors within the past several thousand years, not tens of thousands. That distinction matters. It places the shared ancestry within a historically documented window, rather than somewhere in deep prehistory. The headline figure from this study: more than 70% of the Jewish men, and roughly half of the Arab men studied, inherited their Y chromosomes from the same regional paternal ancestors within the last few thousand years. That genetic pattern lines up with a piece of documented history that predates any DNA test. Some Muslim Arab populations descend in part from Christian and Jewish communities that lived in the southern Levant before regional religious conversions reshaped the area. The genetics, in other words, wasn't contradicting the historical record. It was matching it. This study was reported at a Cold Spring Harbor genetics meeting while regional conflict was actively dominating the news cycle, and coverage at the time explicitly framed the science against that backdrop, for better or worse. 3 separate teams, 3 separate sample sets, the same conclusion. That's when the question shifted from whether there was overlap at all to how deep that overlap actually ran. Doron Behar's 2010 Nature paper, titled The Genome-Wide Structure of the Jewish People, marked a real jump in scale. Where earlier work had focused on single chromosomes, this study moved to full genome bead array genotyping, capturing far more of the genetic picture at once. Behar's team compared 14 Jewish diaspora communities against 69 non-Jewish Old World populations, and 25 of those populations had never before been genotyped for a comparison of this kind. The central finding was striking in its clarity. Most Jewish samples clustered tightly together genetically, and that cluster overlapped most closely with two specific non-Jewish populations, the Druze and Cypriots. That overlap didn't extend as strongly to other Levantine populations included in the same study, and it extended even less to the non-Jewish populations Jewish communities had lived among throughout the diaspora, in Europe, North Africa, and elsewhere. Two populations broke from this pattern in ways the authors flagged directly. Ethiopian Jews, known as Beta Israel, and Indian Jews, the Bene Israel and Kochini communities, clustered genetically with their local non-Jewish neighbors rather than with the broader Jewish cluster, reflecting Ethiopia and Western India respectively. There's a genuinely interesting wrinkle inside that exception. Bene Israel Jews cluster with their Indian neighbors at the whole genome level, and yet they carry a documented paternal genetic link back to the Levant. Two different lines of evidence, drawn from the same population, telling two different parts of the same story. The paper's own summary line put it directly. The results trace the origins of most Jewish diaspora communities to the Levant. Druze is the word worth sitting with for a second, because it shows up again almost every time this kind of data gets rerun. The Druze are a distinct religious and ethnic community, concentrated in the mountains of Lebanon, Syria, and Israel, practicing a closed, non-proselytizing faith that has held its boundaries since the 11th century. Across multiple independent studies, including Behar's 2010 work and earlier Y-chromosome research, the Druze keep being identified as one of the closest population matches to Jewish genetic clusters. Researchers point to a fairly consistent explanation. Like Ashkenazi and Sephardic Jewish communities, the Druze have practiced strict in-group marriage, or endogamy, for roughly 1,000 years. That kind of sustained endogamy tends to preserve an older regional genetic signature rather than gradually diluting it through intermarriage with surrounding populations. Cyprus shows a similar pattern, though for a different underlying reason. As an island, its geographic isolation historically limited how much outside gene flow could enter the population in the first place. The takeaway researchers draw from both cases is consistent. Isolated low intermarriage populations function like genetic time capsules for a region. That's part of why Jewish diaspora communities, often kept isolated by law or custom in whatever country hosted them, still carry a legible Levantine genetic signal after 2,000 years of dispersal. The population that turns out to be genetically closest to world Jewry, across several of these datasets, isn't Jewish, Israeli, or Palestinian at all. It's a small, closed religious minority that most people outside the region have never even heard of. Endogamy explains why the diaspora signal held together across 2 millennia. It doesn't explain what was structuring the region genetically before any of these communities existed in the form we recognize today, and that turns out to be a genuinely different kind of study. Mark Haber's 2013 paper in PLOS Genetics, titled Genome-Wide Diversity in the Levant Reveals Recent Structuring by Culture, arrived with a counterintuitive core finding. The genetic structure visible across the modern Levant is shaped more by religious and cultural history than by simple geography or physical distance. The mechanism Haber's team identified traces back to the 7th century Islamic conquests. Populations across the region that converted to Islam grew more genetically similar to other Muslim populations, spread across a wide geographic area, while communities that didn't convert, Christians, Jews, and Druze among them, retained older, more localized genetic signatures, tied more tightly to their specific place than to any religious bloc. The practical result is almost counterintuitive on its face. Two villages sitting only a few kilometers apart can show more genetic difference from each other than two communities separated by hundreds of kilometers, provided their religious and conversion histories diverged at some point in the intervening centuries. Proximity on a map turns out to matter less than which historical current a community's ancestors were swept into. This finding reframes the entire question this documentary has been building around. The deeper driver in the data isn't a clean two-group split between Jewish and Palestinian populations at all. It's a much older regional population. one that existed long before either modern identity took its current shape, being reshaped repeatedly by waves of conversion and migration across centuries. There's a direct implication for every Y chromosome number covered earlier in this series. The 70 and 82% overlap figures and the language of a shared pool reflect this same underlying cultural structuring process. They aren't describing two genetically sealed populations that simply stayed apart for millennia. If culture reshaped the regional picture that dramatically in just the last 1,400 years, the obvious next question is what the region looked like before any of it, and answering that meant going back to actual bones. Haber and colleagues published in the American Journal of Human Genetics in 2017 what stands as the first ancient whole genomes ever sequenced from Bronze Age Canaanite skeletons. The sample consisted of 5 genomes, drawn from individuals roughly 3,700 years old, excavated in Sidon, Lebanon, once a major Canaanite coastal city-state. Those 5 ancient genomes were sequenced alongside 99 genomes collected from present-day Lebanese volunteers. The central finding established something rare in ancient DNA work: real, measurable genetic continuity. Present-day Lebanese populations derive most of their ancestry directly from this Bronze Age Canaanite-related population, spanning roughly 3,700 years in the same region without a major replacement event breaking that line. The Canaanite ancestry itself wasn't a fixed starting point either. It formed through an earlier mixture between local Neolithic Levantine populations and Eastern migrants genetically related to Chalcolithic-era Iranians, a blending event dated using linkage disequilibrium decay analysis to somewhere between roughly 6,600 and 3,550 years ago. Later, a second and separate pulse of Eurasian-related ancestry entered the region, one that isn't present in either the Bronze Age Canaanite genomes or the earlier Levantine samples, estimated to have arrived somewhere between roughly 3,750 and 2,170 years ago. The significance of this study reaches beyond Lebanon's borders. The Canaanites themselves left almost no surviving textual records of their own. Most of what's known about them historically comes filtered through Egyptian and biblical sources, written by other people, for other purposes. This was the first time Canaanite DNA got to speak directly, on its own terms, about who these people actually were. Sidon answered the Lebanese side of the question. The southern Levant, the land Israel, Gaza, and the West Bank now occupy, needed its own ancient genomes, and those arrived 3 years later. Agranat-Tamir and colleagues published in Cell in 2020 a study titled The Genomic History of the Bronze Age Southern Levant. The sample drew on 73 ancient individuals, extracted from 5 archaeological sites spread across the southern Levant, the specific region covering present-day Israel, Gaza, and the West Bank. The central finding placed both modern populations at the heart of this documentary on the same ancestral foundation. Present-day Jewish groups and present-day Levantine Arabic-speaking groups both show genomes consistent with 50% or more of their ancestry tracing back to people who lived in the Bronze Age Levant, combined with Chalcolithic-era Zagros populations from the Iran and Caucasus border region. This marked the first ancient DNA study to set both groups being compared throughout this series on the same baseline using direct ancient genetic evidence, rather than inference drawn only from modern samples. The study also traced how that Zagros and Caucasus-related gene flow entered the region. It wasn't one single event. It arrived in pulses, continuing from the Intermediate Bronze Age through at least the Middle to Late Bronze Age, a gradual and repeated process rather than a single migration wave. The researchers were direct about the limits of their own data. Both modern groups also carry ancestry components that the available ancient DNA samples can't fully explain. The story isn't finished, and a meaningful stretch of the region's genetic history since the Bronze Age still remains to be sequenced. This 2020 ancient DNA baseline didn't simply confirm the old Y chromosome overlap numbers from 2000. It pushed the shared ancestry story back roughly 3,000 years earlier than any of the original studies had been able to reach. One later meta-analysis took that same ancient baseline and asked an even sharper question: which modern group tracks closer to the actual Iron Age population? A recent systematic review took a wider lens than any single study covered so far, screening 161 separate studies of high-coverage ancient DNA from across the region. Rather than comparing modern populations directly against each other, this analysis used Q-PADM statistical modeling alongside F-statistics, methods built specifically to estimate what proportion of a modern population's genome can be explained as a direct mixture of specific, named, ancient source populations. The comparison point here was the Iron Age Levant, roughly 1200 to 586 BCE, the period covering the biblical era kingdoms of Israel and Judah, among other regional polities. Against that ancient baseline, modern Palestinians showed a primary ancestral core estimated at 81 to 87% direct descent from the Bronze Age Levant population, a figure the review found statistically well supported, with results holding at a significance threshold above 0.05. Among everything covered in this review, it stands as one of the highest in situ regional continuity figures found anywhere in the data. The same review pulled in historical demographic context to frame that number. Ottoman-era records describe a Levant population that was predominantly Arab, Muslim, and Christian, with Jewish population numbers in the region rising gradually only through 19th and 20th century immigration waves. What makes this study matter for the core question running through this whole series is specificity. It's one of the first attempts to attach a precise, sourced percentage to regional genetic continuity for a Palestinian population on its own terms, rather than simply measuring how much overlap exists between two modern groups compared against each other. That's the paternal and whole genome side of the story, settled about as far as current data allows. The maternal side of the Jewish story turned out to be a completely different, much messier picture. Everything covered up to this point has concerned Y-chromosome data, passed from father to son, and autosomal whole genome data, drawn from the entire genetic code. Mitochondrial DNA works differently. It passes only from mother to child, tracing a separate lineage entirely, and it tells its own story, independent of whatever the paternal line shows. Behar and colleagues published an earlier hypothesis in 2006 in the American Journal of Human Genetics. Based on the mitochondrial data available at the time, that paper proposed that the four major Ashkenazi maternal founder lineages traced back to the Near East, mirroring the paternal story that had already taken shape through the Y-chromosome research. Costa and colleagues, led by Martin Richards at the University of Huddersfield, published a 2013 paper in Nature Communications that directly overturned that picture, using full mitochondrial genome sequencing rather than the more limited markers available 7 years earlier. Their finding, the four major Ashkenazi maternal founder lineages, together accounting for roughly 40% of all Ashkenazi mitochondrial DNA variation, trace their ancestry to prehistoric Europe, not the Near East, not the Caucasus. Most of the remaining minor founder lineages showed a similar deep European ancestry pattern running underneath them. The study's own conclusion was stated plainly. The great majority of Ashkenazi maternal lineages were not carried out of the Levant, as had commonly been assumed. They were assimilated within Europe itself, pointing toward large-scale conversion of local European women into early Ashkenazi Jewish communities, generations before the population took the shape researchers study today. That single finding split the field. Not everyone accepted it, and the argument over it is still active. 3 competing accounts of Ashkenazi origin now sit on the table, and they don't agree. Behar's 2006 paper argued for a Near Eastern maternal origin. Costa's 2013 paper argued for a European maternal origin arriving through conversion. And in the same year, Aaron L. Haake proposed something different again, a Caucasus, or Khazar, origin for Ashkenazi Jews broadly, not confined to the maternal line alone. Most later genome-wide studies push back against that third option specifically. Behar's own 2010 autosomal data, along with Haber's broader Levant research, do not support the Khazar or Caucasus hypothesis, even as Costa's separate finding on European maternal ancestry continues to stand on its own. A later editorial survey tracked something worth noting directly. 9 separate genome-based estimates of Ashkenazi Levantine ancestry percentage, published between 2009 and 2017, show a measurable downward trend over time as methods and sample sizes improved. The numbers moved from near-total Levantine estimates in earlier work toward more intermediate figures as the field matured. More recent reanalysis pushes back again in the other direction. Newer work comparing the ratios of absorbed versus founder mitochondrial lineages in Ashkenazi populations argues that the original founding maternal lineages were more plausibly Near Eastern after all. The reasoning Y-chromosome Near Eastern origin is well established at this point, and a split origin model, Near Eastern fathers paired with European mothers, is statistically harder to sustain than a shared Near Eastern origin running through both lines together. The honest framing here is straightforward. This specific sub-question, where exactly Ashkenazi maternal lineages originated, remains a genuinely open, actively contested area of population genetics. That's distinct from the broader Jewish-Palestinian shared ancestry finding, which multiple independent methods now agree on. The most contested number in this entire field isn't the Jewish-Palestinian comparison at all. It's a fight entirely within Jewish population genetics, over Ashkenazi maternal lines specifically. With the paternal and ancient DNA case fairly settled, and the maternal case still an open argument, it's worth widening the lens to the other Jewish communities this documentary hasn't covered yet. Nebel and colleagues, in their 2001 paper, produced a specific finding on two communities not yet discussed here. Kurdish and Sephardic Jews were genetically indistinguishable from one another in the Y-chromosome data sampled. Both groups differed slightly, but significantly, from Ashkenazi Jews within the same dataset, a pattern consistent with different degrees of later admixture accumulated along each community's specific migration path. The historical paths themselves diverge sharply. Sephardic ancestry traces through the Iberian Peninsula, right up until the 1492 expulsion scattered that community elsewhere. Kurdish Jewish communities, by contrast, remained within the Middle East throughout their history, settled across present-day Iraq, Iran, and Turkey, without ever leaving the broader region. Because Kurdish Jewish communities never left, researchers treat their genetic profile as one of the closer surviving approximations of the ancestral Middle Eastern Jewish population, before the diaspora scattered other branches out into Europe and elsewhere. Nebel's study offered a proposed explanation for the small divergence seen in the Ashkenazi samples, specifically possible low-level gene flow from surrounding European populations, combined with genetic drift accumulated across centuries of isolation in Europe. That explanation sits separately from, but remains consistent with, the later Costa maternal line finding covered earlier, even though the two studies were measuring entirely different kinds of genetic evidence. Two more branches sit even further from that Middle Eastern baseline, and their genetic story has nothing to do with the Levant at all. Behar's 2010 study produced a direct finding on two more communities. Ethiopian Jews, known as Beta Israel, cluster genetically with their non-Jewish Ethiopian neighbors, not with other world Jewish populations scattered elsewhere. The same pattern holds for Indian Jewish communities. Bene Israel and Kochini Jews cluster with neighboring populations in western India, rather than with the broader Jewish genetic cluster identified earlier in this series. There's a specific wrinkle worth isolating within the Bene Israel case. A documented paternal Y-chromosome genetic link back to the Levant exists within this population, even though their whole genome autosomal profile matches their Indian neighbors far more closely than it matches any other Jewish community worldwide. Researchers interpret these patterns as the result of small founding groups, originally of Levantine origin, who over many centuries intermarried extensively into the local populations surrounding them. Religious and cultural identity persisted across those generations, even as the underlying genome gradually shifted toward the host population around each community. This matters directly for the comparison at the center of this documentary. It demonstrates that Jewish genetic ancestry isn't one uniform signal worldwide. The same religious and cultural identity sits on top of genuinely different underlying ancestry, depending entirely on a given community's specific migration history across the centuries. That variation inside world Jewry is exactly why single-number headlines about Jewish DNA oversimplify what multiple decades of data actually show, and the same caution applies just as directly to the Palestinian side of this comparison. Just as Jewish DNA turned out not to be monolithic, the studies compared throughout this series treated Palestinian and Israeli-Palestinian Arab samples as populations with their own internal structure, not a single undifferentiated genetic block sitting on one side of a two-group comparison. Nebel's 2000 research identified this substructure directly. Two distinct, frequently occurring Y-chromosome haplotypes showed up among Israeli and Palestinian Arab samples. One was spread broadly throughout the region, appearing at roughly 14% frequency. The other concentrated mainly in the northern Galilee area at roughly 8% frequency, a geographically distinct pattern rather than an evenly distributed one. Additional geographic substructuring turned up specifically within the highlands of Samaria and Judea, distinct again from the Galilee pattern already noted. The region's internal genetic map wasn't flat. The J1 haplotype cluster covered earlier, the one accounting for roughly a third of Palestinian Y chromosomes and rarely found in Jewish samples, is itself concentrated unevenly rather than spread uniformly across all Palestinian communities. That uneven distribution is consistent with a later historical migration layer originating from the Arabian Peninsula. layered onto the older regional baseline rather than replacing it. This ties directly back to Haber's 2013 finding on culture outweighing geography. This internal Palestinian structuring likely reflects that same historical process at work. Religious conversion history and local marriage patterns reshaping the genetic map well after the ancient Bronze Age baseline had already been established. With both sides of this comparison now shown to be internally diverse rather than uniform, the honest next step is asking what the disease and tissue marker data adds, because it's been quietly confirming the same regional picture from a completely different angle. HLA, or human leukocyte antigen, is the tissue matching marker system used in transplant medicine, and studies of Jewish and Arab bone marrow transplant patient populations within Israel have examined it directly. These studies found overlapping HLA haplotype frequencies between Jewish and Arab patient populations, a practical, clinically relevant echo of the same shared regional ancestry already visible in the Y-chromosome and autosomal data covered earlier in this documentary. Separately, decades of Jewish population genetics research, documented as far back as physicians studying newly arrived diaspora immigrants at Israeli ports of entry, catalog specific recessive genetic conditions concentrated within individual Jewish subcommunities. These diaspora-specific founder effect conditions serve as a direct genetic fingerprint of centuries spent in small, isolated populations across Europe, North Africa, or Central Asia. That isolation is the flip side of the same mechanism discussed earlier regarding the Druze and Cypriot populations, the same forces that preserved an older Levantine signal in low intermarriage communities, also produced these concentrated recessive conditions within specific diaspora branches, depending on where each community settled and how isolated it remained. The reason to include clinical data at all in a documentary otherwise built on Y-chromosome and whole genome studies is straightforward. It demonstrates that the shared ancestry finding running through this entire series isn't confined to one type of genetic marker or one research method. It shows up independently in paternal lineage data, in the Levantine preserved portion of maternal lineage data, in whole genome data, in ancient DNA extracted from actual skeletons, and now in clinical tissue matching data used every day in hospitals, arrived at for entirely practical medical reasons rather than any historical inquiry at all. Independent confirmation across 5 different kinds of genetic evidence is about as strong as population genetics gets, which raises the obvious question of why this research doesn't get talked about more. Despite 2.5 decades of consistent findings across multiple independent teams and methods, this research rarely enters mainstream conversation about the region. There are specific reasons for that gap, and they're worth naming directly. The first reason: genetic ancestry data gets weaponized on both sides of unrelated political arguments, used incorrectly to claim territorial legitimacy in either direction, even though not one of the studies cited throughout this documentary makes or supports that kind of claim. The data and the political use of the data are two separate things, and they get conflated constantly. The second reason: population genetics results are probabilistic and population level, not individual. A shared ancestry percentage describes historical population movement across centuries. It says nothing about any single person's identity, rights, or legal status. The third reason: some of the loudest public claims circulating about this topic online come from outside peer-reviewed science entirely, including at least one widely circulated but scientifically unsupported paper on Palestinian and Jewish ancestry from the 2000s, later heavily criticized within the genetics literature itself for serious methodological problems. Bad science travels faster online than the corrections that follow it. The fourth reason is more mundane and purely technical. Ancient DNA work in this particular region is genuinely difficult. Hot, sometimes wet burial environments degrade ancient DNA faster than cooler climates do elsewhere in the world, which is part of why the Bronze Age Canaanite and Southern Levant genomes took until 2017 and 2020 respectively to sequence at all, decades after the original Y-chromosome studies had already begun. The scientific consensus on shared ancestry has been remarkably stable since 2000. It's the public conversation around that same data that has stayed unstable. None of that instability changes what 2.5 decades of independent labs, methods, and sample sets keep landing on. Laid end to end, the chronological arc runs in a specific, named order. Santachiara Benirachetti's 1993 work offered the earliest hint. Hamer's 2000 worldwide Y-chromosome study, published in PNAS, followed. Nebel's 2000 and 2001 papers established the 70 and 82% shared pool figures in the American Journal of Human Genetics. Oppenheim's research added the 3 haplogroup clustering result. Behar's 2010 whole genome study, published in Nature, surfaced the Druze and Cypriot overlap. Haber's 2013 paper established that culture, not geography, drives the region's genetic structure. Costa's 2013 paper identified the Ashkenazi maternal European finding. Haber's 2017 work sequenced the first ancient Canaanite genomes. Agranat-Tamir's 2020 paper, in Cell, sequenced the ancient southern Levant genomes. And most recently, a meta-analysis screening 161 studies produced the 81 to 87% Palestinian continuity figure. What changed across that timeline wasn't the underlying question. It was the method. Single-marker Y-chromosome comparisons gave way to whole-genome autosomal data, which gave way to full ancient DNA extracted directly from Bronze Age and Iron Age skeletons. Each new method, arriving independently and often decades apart, landed on a version of the same core finding. Stated as precisely as the data supports, without overclaiming, modern Jewish and Palestinian populations share a substantial, well-documented degree of common regional ancestry rooted in the ancient Levant, layered with distinct later histories. Diaspora dispersal and partial conversion-driven admixture shaped one side. Continuous regional presence with its own internal migration layers, including the Arabian Peninsula-linked J1 cluster, shaped the other. What the data does not claim is equally clear. It does not determine modern political, legal, or territorial questions, and no cited study frames its findings that way. The one genuinely unresolved thread running through the whole documentary is the exact origin of Ashkenazi maternal lineages, specifically, still an active argument as of the most recent papers. Every one of those studies started with a testable question and let the lab work answer it, which is worth sitting with, one last time, before closing this out. Return, for a moment, to that opening scene. The year 2000, Cold Spring Harbor, New York. Researchers reading Y-chromosome data aloud at a genetics conference while regional conflict dominated the news cycle just outside the room. The through line running from that moment to now can be stated in one clean sentence. Independent teams, working across different decades and different lab techniques, kept arriving at the same regional ancestry story, confirmed again as recently as ancient genomes sequenced directly from Bronze Age skeletons. That said, the genuinely open scientific questions deserve honest acknowledgement rather than a tidy bow. Ashkenazi maternal origins remain chief among them, still actively argued over in the literature as of the most recent papers covered in this documentary. This isn't a field presenting itself as fully closed, and it shouldn't be treated that way. What a story like this actually demonstrates is something about the scientific method itself, separate from any particular finding. A politically loaded question got a stable, replicated, methodologically diverse answer precisely because researchers kept testing it with better and better tools, generation after generation of technology, instead of assuming the answer in advance and going looking for data to support it. After 2.5 decades of consistent data from completely independent labs, using completely different techniques, what would it actually take to change this particular scientific conclusion at this point? Drop into the comments and share which finding in this video surprised you most.